Welcome everybody for today's talk in structural biology. Today's topic is a bit different than the previous ones. This topic is a bit closer to the research interests and activities of my own research group, but nevertheless I will give you, hopefully I give you a fair introduction in structural biology and what it can do with understanding biological photoreception. Now, why is biological photoreception important? Everybody has a reason why his or her topic is important. When you look on this slide here, you see our sun with its usual flares, and as you all know, the sun is the ultimate source of all the energy, almost all of the energy that we have here on Earth available for terrestrial life. You see the huge numbers that we do have. About a kilowatt per square meter is the flux that we get on a sunny day. About 1% of the incoming light of the sun is converted by organisms in biological matter in the process called photosynthesis. We know that about 220 billion tons per year of carbon are fixated by these organisms per year, and also some part of this carbon that is being fixated by life is getting sequestered. So about 3 gigatons, 3 billion tons per year get into the sediments and will not be released to the atmosphere unless, of course, humans dig it out of the earth and burn it for something. And as you should all know, there are many important CO2 fixation pathways and the pathways for the formation of sugars and so on. Photosynthesis, crucial central aspect of biochemistry and biophysics. So when we look on our planet Earth from the space, as you see here, you see that it's not only blue, but it's also green because of life, of actually plant life on the surface because of photosynthesis. You see that here, for example, some algal blooms in the Baltic Sea. So everybody welcome to see this summertime again and again. But also sometimes you get different colors than green or this green bluish. You get colors like these ones here in a salt lake where actually a green part of the lake is separated by this reddish part of the salt lake, the Great Salt Lake in Utah. And in this lake there are red algae existing and also under very high concentrations haloarchaea, which give a different color to that. And you see that from space. And here on the right hand side you see bioluminescence in front of the Horn of Africa, in front of Somali, that is made by bacteria which produce light in the oceans. And we know most of the structural features of that cause these processes. We know about the photoreaction center from the pioneering works of Hartmut Michel and Johann Deisenhofer, but also of later work from other authors on more complex systems. We know about the way how simple organisms use this simple protein, bacteriorhabdin, to generate a proton motive force to fixate energy along a biomembrane potential. And we also know a little bit about bioluminescence because it's also very interesting for chemistry because there are some photochemical reactions happening. So when we consider what's going on Earth in terms of electromagnetic radiation, you see that actually we get broad band from the infrared till the near UV region. Quite luckily, most of the UV ultraviolet radiation is filtered out by our atmosphere, by the ozone layer. Only a minor part is reaching the surface of our Earth. The majority of energy comes here in the visible range, and that visible range, a little bit more from the visible range to the UV range and to the infrared range, that is a range that can be used for triggering chemical reactions. That means not only vibrational changes, which cannot change chemistry on itself, mostly at least, but real rearrangements, electron transfer, the reactions that actually can fix energy that is coming by incoming photons. So when considering now why light is so important, not only in terms of being an energy source for life, but also because it tells a lot about life, where it is and what its environment actually is. So you see that, for example, here, when you have light shining on our landmass, it's on every part of the planet the same, but it's at least on a sunny day. When you look into water, where actually life originated, you see that the distribution of light changes with the depth in the water column. So here at 200 meters, you only see a little bit bluish light coming through and nothing else from red or from purple. It's different, can be different here in coastal waters, for example. When you have a lot of mud and other kind of scattering matter in it, you change this kind of spectrum considerably. That is being measured at any kind of given position here in this water column. That is telling organisms actually quite a bit, where they are in the water column. And remember, they don't have ways, to determine their place in life or in the water. So the biological effects of light are multitude: photosynthesis, damage, and sensing. And there are also some other biochemical reactions. Most important is biosynthesis of vitamin D in our skin, because that's an essential component of us. Otherwise, you get rachitis. Chlorophyll biosynthesis is driven by light. Interestingly, there's a light-dependent reaction. And I will not tell about anything about photosynthesis in the following slides. Although this is a very interesting topic, but we could fill hours with this as well. So I will tell a little bit about sensing. Yes, how actually light is sensed, what structural biology can tell us about the kind of mechanisms. And I will not go for all the families. We have to restrict ourselves to some families only. So first of all, why is it so fascinating? Or what's so fantastic about photoreceptors? So you all know that these photoreceptors are proteins, and these proteins, in order to sense light, they have to have a chromophore, something that is absorbing the light. Mostly that cannot be done by the side chains of the proteins. There's only one exception. The urea receptor UVR8 is using tryptophan chains to sense ultraviolet radiation. But all other photoreceptors require a chromophore that is inbuilt. So the light reaction is very, very fast, femtoseconds to picoseconds. So that's something for ultra-fast spectroscopy for ultra-fast spectroscopists with whom you have to work in order to get out something about the chemical basic photochemistry of this reaction. So you see that's happening within a picosecond. The fancy part about the biological photoreceptors is that the story does not stop at this moment, but it just starts. Because we have much, much slower reactions that go in the microseconds range and which are triggering proton transfer. We will see examples. Electron transfer inside the protein or between protein subunits, or even some conformational changes being triggered in the protein itself. And then on an even longer time range, in the microseconds to second time range, we really get actually our photoreceptor in a biologically meaningful state where it can actually do something, give some signal, make some biology. Because biology is not happening on the femtosecond level, it's happening more on the microsecond to second level, because most reactions are slower. To bind, for example, an interaction partner, that takes microseconds to milliseconds. So we have an unfolding, refolding reaction, binding, and dissociation. And good photoreceptor is capable with, after having absorbed the light and making its primary photoreaction, to have almost a 100% success rate to form the signaling state at the very end. And that is away 6 to 9 orders of time. So there are several families known. We assume nowadays that we almost know all kind of photoreceptor families. They're here shown for the prokaryotes, for the bacterial organisms. An example are the plants here. Here you see the plants. Plants have many, many photoreceptors. Clearly, they cannot walk away. They have to sense the kind of light conditions they are being, they owe to their place. And they have to react in some way to this, and you see there are several loft domains, phototropins, cryptochromes, UV receptors, phytochromes, and here in the midst, in the green range, there's almost nothing, because as you can imagine, it doesn't make sense to have something in here in that area, because there is actually the point where the photosynthesis is also not working anyway. The green stuff is actually the light that we mostly see from the plants that is not absorbed. Yes, it's not meaningful for our information, but for us it's important. We have green light receptors, retinals, or rhodopsins, retinal-dependent rhodopsins. So when you consider these kind of 7, 8 families, that we do have nowadays, I think it's 9 families, we have here the phytochromes that use a linear tetrapyrrole that is using a cis-trans isomerization around a single double bond, not around the other double bonds, but just a single double bond, 13, 15 double bond. We have rhodopsins, which use retinal, which is a linear polyene, which also isomerize usually around a single double bond, usually this here or this one, depending whether it's animal-like rhodopsin or prokaryotic rhodopsin. Then we have xanthopsins, which use a very simple compound for hydroxycinnamic acid. I don't know why this is missing here. Some bacteria. Cryptochromes, love domains, bluff domains, they all have in common that they use flavins for electron transfer, for adduct formation, for hydrogen bond changes. Okay, there's actually, it's a pull down again. I hate this kind of slides. So these are all the families that we have, and we know the basic mechanisms of most of them. Let's make first a look on cis-trans isomerization, because it's something for chemists like you that should be close to your love for organic chemistry, as I have it sometimes. So cis-trans isomerization can be triggered by light, of course. One of the important aspects is in photoreceptors, when you have several double bonds available where isomerization can happen, it's only a single double bond that is being allowed in photoreceptors to go the isomerization. So when you buy, for example, retinal and you illuminate it here on just normal light here or sunshine outside, it starts to isomerize at every single double bond here where it can isomerize, but not in the photoreceptor. It's fixed. It's just this single double bond, and that one for the animal rhodopsins. Likewise, the phytochromes, and okay, with this 4-hydroxycinnamic acid, there's only one double bond. We don't have selection about what's going on there. So when looking on the most simple photoreceptor that we do know nowadays, let's call it something like the lysozyme of photoreceptors. It's called the photoactive yellow protein, PYP. It's from some bacterium. It's very small, 14 kilodalton, and it has this super simple chromophore, this 4-hydroxycinnamic acid that we have seen before, that is bound via its carboxyl group as a cyroester to some single cysteine residue in PYP. Funny thing is, it's somehow the lysozyme of photoreceptors, so everybody can study it. It's done for a lot of, to establish different technologies, also high-end technologies. But the problem is the biological function in that particular organism is not well understood. So in other bacteria, it's often fused to some histidine kinase, and then it has something to do with two component signaling, where the light is then the incoming signal, and the histidine kinase changes its phosphorylation activity on other protein targets. So that's about PYP. But why we chose PYP? Because we can learn quite a lot of simple things how these kind of proteins are studied. First of all, how are our biophysics friends studying these kind of proteins? They use pump probe absorption, or sometimes also pump probe fluorescence spectroscopy, to measure the temporal dynamics of the photoreceptor after it has been hit by a photon. So the setup should be known to some of you, most of you, at least when you listen to our friends from physical chemistry, who should do this, for example, like the Weitzel group, but they work with hydrogen chloride or some samples like that. I don't know what they're working with. So the sample light here is being here used to probe the sample, and the detector is collecting the spectrum, for example, and a laser pulse, a short pulse, is triggering the probe, and just by the delay between when you flash with the laser and you measure with the sample light, you get then a spectrum. And you can record this spectrum here, after being hit. Yes, in the time here it's log time in microseconds, so log 6. I think it's the log to 10 base. Yes, So they measure here some to minus base from microseconds to seconds. Probably you see some changes here in the absorption evolving around. So this is a pretty complex technology. When you go to femto to picosecond spectroscopy, that's done by some specialized laboratories, for example, the laboratory of Josef Wachtfeitel in Frankfurt or Pascal Plaza in Paris, or some other specialized labs who can do that kind of experiments on these delicate samples, because you remember proteins are not like solids. So when you do solid state chemistry, proteins like to be in the buffer, being cooled a bit. They are not so stable to get this kind of data. So these data, usually when everything goes fine, will give you then a sequence of intermediates that are formed during the photoreaction. In this case, it's a cycle, because this PYP, this photoactive yellow protein, when it's being hit by light, forms first the hot ground state that decomposes in picoseconds to this PR510 state, then nanoseconds to PR565, and then back to some bluish-like state here, and that takes milliseconds, relatively long time when you compare it to the other time scales, to return to the initial state. So that is a photocycle. Not all photoreceptors undergo photocycles. Our rhodopsin in our eye is not undergoing a photocycle. It's a one-headed reaction. It starts from the initial state, it forms a signaling state, and then it decomposes and releases the chromophore from the photoreceptor, and the photoreceptor has to take up a fresh chromophore for becoming restored. So not all photoreceptors are undergoing photocycles. Now the problem is, of course, how to study photoreceptors in general. So clearly you can say it's nice when I have a photocycle like this. I can measure this maybe with an ultra-fast spectroscopist. But there are some other techniques, especially when you want to learn something about structures. We cannot, at least till a few years ago, we could not record structures in these kind of short time ranges. We couldn't resolve 10 years ago structures just a picosecond after the laser pulse came in. So what we did, was doing some experiments like taking the protein, illuminating it by light, but keeping the temperature low, minus 150 degree or minus 180 degree, and then warming it up, stepwise. And that can be done with whole crystals. So you illuminate a crystal by laser light at cryogenic temperatures. Everything gets being converted according to the quantum yield. And then you can make the target temperature, and according to the activation energy barrier that has to be overcome during this kind of photoreaction cycles, the system goes maybe over the first barrier but gets stuck in the first local minimum, for example, in one of these intermediates. Then you warm it up a bit more and maybe it can go to the second minimum. And when you have good luck, that has something to do with the photocycles measured by our ultra-fast spectroscopy people. Now the answer is yes, it usually has something to do with the kind of real in-solution dynamics, of your photoreceptor, but sometimes there are deviations. For example, you see here additional intermediates and wavelength numbers coming up. The wavelength numbers are the lambda max values, the maximal absorption of the photoreceptor when probing it with light. You see they are different than the ones here recorded at room temperature. For example, this signaling state is not exactly the same like that signaling state. And when you do that in a simple lab like us, in a photospectrometer, you make maybe steady state, you switch on the light, you get an equilibrium between the ground state, the initial state, and the long-lived intermediate state. And sometimes you can see this and you can measure that also in a standard spectrophotometer to see a photosteady state. So please remember, when we're having photoreceptors, they form transient intermediates. You can trap them sometimes, but by low temperature, but they give sometimes different kind of results. You can even study them under steady state conditions, but the steady state conditions give you only the distribution of the most stable intermediate in the initial state usually, not the other intermediates in between. So how is actually structural conformation change of the chromophore transmitted to conformational change? So first of all, when we consider here the chromophore binding site of PYP, here shown in yellow, and here shown the chemical structure to this, we see that it's making strong hydrogen bonds with surrounding amino acid residues. What's interesting, the cinnamic acid, the hydroxycinnamic acid, is deprotonated in solution. pK is at 8.8. Is deprotonated, so the proton is not residing here on the phenolic hydroxy group, but it's residing instead on the glutamic acid residue, glutamate 46, which are being close to that. That's pretty interesting. That means, pK was going for 2.7 in the protein. Mix it up. So actually, it's residing on the glutamate. So that's somehow contradictory to intuition, because usually you would expect, that the chromophore is protonated and the glutamate is deprotonated, because these are the in solution values for the chromophore and for glutamic acid that you can measure. But inside these kind of proteins, there are strong hydrogen bonding networks and they are not necessarily following the energetics in solution. So what's happening? And we can predict that what's happening now. Here's some positive charge, of an arginine residue. Here's another tyrosine residue, making some hydrogen bond. So what's happening is that actually this single double bond will isomerize, and that hydrogen bond will be disrupted, and that proton transfer will start from the glutamic acid to the chromophore. So you see that here in the overview, ground state isomerization starts, protonation of the chromophore happens, and structure change is triggered, and the whole system then finally recovers. So there have been done cryotrap structures in the past, yes, for PYP, end of the 90s. For example, this structure here at 110 Kelvin, cryotrapping by continuous illumination. I think that was a science paper that was claiming somehow status that was not found by other people. You see the difference electron density. Difference electron density is telling you conformational changes. So a difference electron density is calculated by taking the diffraction data from state 1, dark, and the diffraction data from state 2, illuminated, and subtracting them from each other and using the phase information and calculating then difference maps. And the difference maps is telling you where are atoms moving to. So here in blue, that is indicating that, in blue, yes, it's fine. In blue, it's, no, in red. Sorry, I'm probably going to get mixed up. Well, I should have written the color code here to this, yes, In red, it's indicating where density disappears, yes, So here the atoms do disappear, and here in green bluish, electron density appears. And this is reflecting the isomerization at this position here from the trans to the cis double bond. You see also other changes happening here. For example, it's a hydroxy group. It's also moving away a little bit the tyrosine residue in this cryotrap structure. Please remember, it's cryotrap. It cannot move a lot because everything else is a frozen matrix. So one can ask, what is so special about photoreceptors, and I think one of the very simple ideas, having a few years ago was, one can ask, why are some proteins photoreceptors and others are not? We know other proteins which have maybe similar cofactors bound, but they are not acting as a photoreceptor. They absorb maybe light. Yes, green fluorescent protein, for example, they absorb light. They fluorescence back some of the energy, it's gone. But there's no conformation change happening. Nothing is being done on a long-term scale. There's no directionality of the reactions happening. And the idea was, to compare it with some enzymatic reactions where you also say, you have to get some directionality in the reaction also of the conformational change, maybe you stabilize the transition states, by after the photo excitation has happened, the first photo product has been formed, and then actually, because then you have a stabilization here, for example, here for this transition state, the P state, can easily cross over to the main reaction coordinate and then go on and produce and finally the product. So PYP has a pretty good quantum yield. 38% of all photons that are absorbed cause trans to cis isomerization, cause a signal formation. In bacteriorhodopsin, we have about 64%, and in phytochromes, for some reason, they're pretty inefficient, about 15%, but not more. So phytochromes are certainly not the perfect receptors for sensing very low levels of light. So they are something like our rhodopsins. They have fantastic quantum yields, yes, but not the phytochromes. Okay, how can we study with time-resolved crystallography these kind of structural changes? The first idea was, to use our synchrotrons as we have it here, and to use them in a very perverse mode to not fill them with as many electrons as possible, which are going here in cycle and produce a lot of radiation that can be used for crystallography or other material science studies, but just to put a single bunch of electrons in and let them circulate here around the ring. So it's about 1 kilometer that has to go, and you can imagine that is producing a frequency train of X-ray pulses, 100 picosecond X-ray pulses, which are separated by 0.5 nanoseconds, because that's the time it takes again for the electron pulse to come here back to our position. So one can generate X-ray pulses with a short time duration and make some diffraction experiments, so-called lower diffraction experiments, where you use all the kind of wavelengths which are in these X-rays and produces super complex diffraction patterns, which Max von Laue has observed and actually explained how they are formed, and calculate electron densities with this. With this one could do the first structures, but only when the whole synchrotron was just working for your single experiment. Here are some example from Keith Moffat's lab, mostly done at the synchrotron in Chicago. He made a movie, yes, ranging from 0.5 nanoseconds to 4 seconds for the photoactive yellow protein. And you see here densities coming up and down. So you have to see here is a time frame, 64 nanoseconds, 120, and so on. And you see there's a lot of changes, but after some time, the different density disappears because now the system is resetting itself back to the initial state, and now it's flash. We have the maximum of change now, and then all the kind of intermediates are being formed and try to wiggle around to reform actually the initial state back. So that was the first film of protein. Now we have much better movies of this protein with a much better time resolution. Now from 2012, we are now in a time resolution here of about 100 picoseconds. That is better than we had before. Maybe let's the movie here. You see here now in a color-coded way how density appears and disappears, and we see how the whole chromophore actually is changing its conformation during this illumination pattern. So that's about what one can do at synchrotrons. Here you see the storage ring, the X-ray pulse. Before you pulse, like with the pump probe experiment we have seen before, you do the laser flash. Your observation pulse is this time not light, but X-ray. You have a crystal that is being transformed during that time, that produce a diffraction image, and you record all these kind of diffraction images, and you just change the time delay in the pump delay experiment to get this movie assembled. Pretty cool, pretty complex work to do so. Took years of work to produce this kind of movies. So we now know much better, how actually this protein is working. We see here this photocycle and also the approximated quantum yields, So the first state that is formed after the excited ground state, that is on the S1 level, is this PR0. Yes, that it's rapidly formed. It has a 90-degree tilt. Let's maybe look whether we see this here. So we see this here, and then the first intermediate, next intermediate coming up here. and then sucks, it's gone perpendicular to that, you know, and everything changes in this protein. And here something more. But of course, the biologists would say, how is the signaling state looking like? How is this so-called PB something looking like? Now there comes one point about it, and that's it, that actually these experiments shown before are all done in crystals. Crystals are not necessarily telling you the truth about the system's behavior. So you have to use sometimes other techniques to know about what's going on. For example, infrared spectroscopy can tell you quite a lot. Infrared spectroscopy, as you should know from your physical chemistry education, is telling you something about vibrational states. The vibrational states are very sensitive to the chemical environment in which actually these bonds or groups are being located. For example, you see here on the right-hand side a peptide bond. Yes, the signature infrared band for the peptide bond. You see, it's different, you know, the form and the precise position of the spectrum for this peptide band, whether it's in an alpha helical context or in a beta sheet context, or whether it's something else, something different. So these kind of vibrational spectra can tell you something about conformational changes within the protein. It's very sensitive, especially sensitive for beta sheets. And you can do this kind of experiments with most step scan FTIR machines available. What comes out when you do FTIR spectroscopy with PYP? So the funny thing is, you know, when you look what's happening here in the black line. So first of all, these are difference fluorescence transform infrared spectra. So the peaks going here from the 0 line up and down, yes, you know, and some of them are assigned because they are calculated between the lit state and the dark state, between the signaling state, this PB-like state, and the PG state. So you see there are a lot of peaks. Some of them could be assigned. Some like here the chromophore where it's located and the double bond and the glutamic acid residue. But what you see, what's the difference here is when you make the same experiment in solution, you have a gigantic change here for the peptide bonds that is indicating that a lot of secondary structure that is in a beta sheet-like conformation is somehow disappearing. And this is not reproduced by the protein when it's being in the crystals. The rest looks fine, more or less fine. But this one here looks really very strange and different, telling you that something is odd, you know. Also the kinetics is different for the structural transitions that you can measure here, you know, for the protein, whether it's in crystals or in solution. It's much faster, I think it was faster in the crystals for some very strange reason. The reason why this is happening is because the crystal structures are all wrong about the signaling state. We do know from NMR spectroscopy that in the signaling state, this protein almost completely, or at least major part of the protein, do unfold an unfolding reaction. So they lose their structure. The former strands start to splay out here in long loopy or disordered regions. And the chromophore is covalently still bound to the protein. It does not get lost by this. And then after it starts to re-isomerize, the protein starts to refold around the chromophore. And that is probably the reason why actually the signaling state, you know, of PYP and the related proteins has more or less to do with the unfolded state the protein transiently takes when being hit by a photon. And also to remind you about the time ranges that we are here, you know, so the photoreception goes very fast, you know. Here you can do quantum chemical calculations when you like to do so, yes, because these are calculations that give you some proper results and predictions for the behavior, but everything else it's very hard to simulate to get it on because it's so slow, yes. Even with molecular dynamics simulations that are run for microseconds, you do not come mostly into this kind of unfolding reactions here. You know, you have to get to the real signaling state of this photoreceptor. So that was an introduction to a simple photoreceptor. Let's take something else. Second photoreceptor, different system, but still isomerization. First of all, I like old slides. This is an old slide, you know, I think I got from Dieter Oesterheld some time ago, you know, showing you some of these kind of marine salt production facilities, you know, where you just evaporate seawater, you know, and the remnants are just here the white salt, which you can then sell as very precious, nice marine salt for for cooking. Yes. So what happens during that kind of water evaporation is, you know, that first of all, you know, water evaporates. Here there's water, it starts to evaporate, then some green algae start to grow up like hell, because they are the only ones who can live in salt conditions up to about 2 molar sodium chloride. And they live, I think, between half molar and 2 molar, and then they start to die because it's also then too much for them, you know, too much salt. And then everything becomes purple. And sometimes you see that when you look for some of these kind of some purple salts. Sometimes it's from metal, sometimes it's of biological origin. You know, it gets purple, and these are these bacteria, these haloarchaea, which we see here. You can even trap them in salt crystals, you know. They are still alive, yes, you know, so you can get them out here. Some industrial production facility where you have the same kind of color change happening. So that's therefore not a big reason that Walter Stocanius, because that facility was here close to San Francisco. Walter Stocanius and Dieter Oesterheld resolved the puzzle about this kind of organism, why it's so strangely colored, and they found there's a protein called Photoreception that is pumping protons out of the cell when being hit by light. And the proton gradient that is being formed up across the membrane can then be used to generate ATP. So you see that's a really old slide with all this kind of dust here on it. Yeah, generates ATP, and also can move the flagellum, because the flagella are also driven by a proton gradient. But not anything else. It cannot make reduction equivalents like our our normal photosynthesis can do. So actually, these organisms depend on some. So they are heterotrophic. They require at least some dead organic matter to live on it. But they can produce most of the ATP just by this light reaction. And you see this here. You can even make some artificial systems. You know, chemists like to do that, or nanobiologists. Nanotechnologists to make some synthetic system where they couple these two components in artificial systems, Photoreception and ATP synthase or whatsoever. So this protein is part of a large family. These haloarchaeal rhodopsins are not only occurring in haloarchaea, but also in bacteria and also in fungi. They are probably responsible for maybe up to 10% of all the kind of light absorption by life, you know, because most of them use it like Photoreception for pumping protons to make proton gradient across the membrane. But you know, for example, in haloarchaea, we have the interesting situation that we have also some other proteins which are very related, which also have the same chromophore bound, the retinal chromophore here. For example, halorhodopsin that pumps chloride anions from outside into the cytosol of the haloarchaeon. Very interesting reaction. It pumps chlorides. And we have, of course, also something called sensorhodopsins. It gives a signal to the signal transduction machinery. So why is it pumping chlorides in? Actually, the current explanation is always that even for growing your volume in these kind of hyperosmolar areas, environments, you have to use energy. You have to use energy to get that somehow counterbalanced, you know, and get other ions then drawn in as well. Potassium ions mostly in this case. So you import chloride ions, but then you also import potassium ions passively with you, because there's some channel in the membrane. Proteins are pretty simple. 7 transmembrane helices, similar like in rhodopsins of us. Our rhodopsins have a different kind of structure, although they have the chromophore attached to a single lysine residue in helix number 7, counted from the N terminus. So that's common. So what's a bit different here with our Bacteriorhabdin is that in the transmembrane regions, there are two charged amino acids. And you see already one point about membrane proteins, about which I wanted to talk today anyway, but now we see that you have usually only hydrophobic amino acids in the transmembrane regions and not polar residues like these two aspartates. Maybe there's one more aspartate here, yes. That's also a function. So all these kind of charged residues inside being part of transmembrane adhesives have mostly to do some function, have to fulfill some function. For example, this lysine is of course a coupling partner with the retina. So the protein is very simple to study. That's the reason why Dieter Oesterheld studied it for about 40 years. So one can see this very nicely with a simple pH meter when one is having a bacterial suspension and putting a pH meter in it and switching on the light, the medium starts to become acidic. Very funny, yes, because it pops a lot of protons out. But you can also see that the bacteria change their color a bit, or when you purify the protein anyway, that they change the color from this purple way to this yellowish way, at least when you have the right mutants, which are long-lived enough. So you can study the whole process in the cell, in the protein, and whatsoever. So Bacteriorhabdin is the lysozyme of the membrane proteins. Every kind of technique that has been invented so far in life sciences and structural biology has been applied to Bacteriorhabdin. You tell me one technology, I'll find you the paper for that, because Bacteriorhabdin is something. When it works with this, it should always, when there's a new technique, it should always work somehow with Bacteriorhabdin. So about pumping, only one thing. In order to pump something, you have to fulfill a photocycle, right? Yes, that's the same case, as we have seen with PYP. It has to return to the initial state, here called BR570, in halodopsin HR580. The first reaction is, of course, the isomerization reaction, because that's what the light is doing, yes, Then there's a point, when there's a transfer reaction happening, so the release or the acceptance of that thing that you like to transport, proton or chloride. And when is the change of the accessibility happening, for that is to be transported, So this is called here a switch. And in this simple model, you can predict there are only certain combinations allowed and other combinations are not allowed. And I think most of the allowed combinations here, this very simple scheme, have been also observed with related pumps, doing the way. But you see here, these two acidic residues that you have mentioned before, they are the key partners here. D85, aspartate at position 85, is releasing the proton to the extracellular side. Aspartate 96, it's on the cytosolic side of the protein, is accepting the next proton, And here in halodopsin, everything is a bit different. Yes, there's a threonine residue, yes, instead of some combination with an arginine residue. And there's some combination always here, threonine, arginine, that is doing this kind of reaction. So that's vectorial transfer, So here's the overview, yes, of this halodopsin. Small protein, 250 amino acids, I like this halophilic archaea, they're not bacteria, they're archaea. They live in 4 molar sodium chloride. Try to make a glass of 4 molar sodium chloride. You fill up your glass, at least half of that, with your sodium chloride. Then you start to pour water in it, and then you get a saturated sodium chloride solution. And don't drink this, because then you will die. Yes, so that is actually the condition where this kind of archaea are actually living. And the direction of proton transfer occurs because, of the isomerization reaction here. Unfortunately, this is from a textbook, so they forget always the double bonds here. So this bond is isomerizing, this one. And by that, it is moving the nitrogen, the Schiff base, from the cytosolic side to the extracellular side in the protein. It's just in the midst of the protein. It's just swapping its orientation. And when it's swapping its orientation, it lets its proton to the aspartate 85. So here the M state that is formed, this intermediate state is then deprotonated. And now the proton can follow on here a chain that is made up of water and of some arginine residue and of some surface exposed glutamic acid residues to follow up this chain just by the Grotius exchange mechanism. And unfortunately, I don't have my slide board with me. I would like to explain the Grotius exchange mechanism in water, because it's a fancy mechanism. Maybe at the end, when we can come for some questions, I take some picture out of the internet. So it's not the same proton that is getting out here. It just makes a hydrogen bond with this oxygen that is releasing this proton then to this water molecule. Yes, that one is releasing into another water molecule that's not shown here. Then this one to this one, just by swapping between the bonded level and the hydrogen bonded level. Proton transfer in watery conditions is extremely fast and also is used up here in this kind of pump. So what about sensorodopsins? Make it short. Sensorodopsins, something for general knowledge of human that you know a little bit more about, also some basics, actually relevant for microbiology. So you see what these kind of haloi here are doing. When you illuminate ultraviolet light, on a cover slip, they try to evade this, to avoid this. So they swim away. They accumulate here in the red area where they feel happy. They don't like this blue ultraviolet light. They hate it, yes. So they see the blue light. They see it really. And what happens when you put on white light? Then they migrate to the white light. So there is a photophobic reaction. They try to avoid light. At least this kind of blue light, ultraviolet light, and a phototactic response. They try to migrate to this position, where they see it. And it's mostly in the green area, where they go to. So that's interesting. So that means these kind of simple organisms can see different colors. So they are like us. We can also see different colors. But I think a cow cannot see different colors. So there's other bacteria using phototactic reactions here, for example, bacteria who spread up over a spectrum, being made up by a prism, and incoming white light and different colors. Or here you see some bacterium that is migrating to the light source by growing in that direction. Yes, a lot of these kind of reactions are known. So in our case of our haloi here, and many other bacteria, and actually they use the classical phototactic for phototactic classical mechanisms, the so-called biased random walk. And you see, today we make also some excursion to something also very interesting, not only everything the structures directly. Biased random walk, what is it? Just imagine you're completely drunken. When you are completely drunken, you either fall to the left or to the right. When the likelihood is equal, yes, to fall to the left or to the right, or to make a step to the left or to the right, actually the likelihood is that actually the distribution where you can be found after maybe 1,000 times, I think, is still in the center. So when you bias your walk, you fall 70% of all your cases to the left and 30% all to the right side. Clearly, you move to the left side over time. And that's the same mechanism is used here by these kind of small organisms, by this kind of bacteria. They swim in one direction. Then there's a built-in time trigger, where they actually reorient themselves. The question is a concentration of, or here in this case, light intensity now higher or lower than it was before when I started my outrun. So depending on this, they actually tumble around, go in a different direction, and you see the lengths here when they start to change when they start to tumble around and by find some random direction to swim now, is proportional to the change of the light intensity. So here light intensity change is continuously happening. So this is a long walk before tumbling occurs. Here light intensity is only to a little bit changing. So tumbling occurs earlier. Here it's even worse. Actually, the light intensity decreases, so it's even earlier, so it tumbles here around. You do this a lot of times. You come to your light source. Here you see this here for an incoming bacterium. swimming to such a spot where some light is put in, you know, for phototactic response. Or here that was for photophobic response, you know, where the blue light is photophobic and the orange light is phototactic. So as you may have heard in biochemistry 2 or 3 maybe, I don't know who is now reading this, you know, this is classical two-component signaling. Gerd Bang is interested in some part of the flagellar motor. I think he was interested in that, at least, you know. The coupling components are always the same, KW, KA, KY, and KY goes to the flagellar motor, you know, and you just exchange chemotaxis receptor like Escherichia coli has it with that receptor, and then you have light reception. And that uses the same pathway to produce this kind of reaction. And in our vision, we have a G protein coupled receptor, as we talked last time. Yes, you know, remember? So our retina is not walking away. We think with our brain first what we are doing. So bias run and walk is also causing chemotaxis. That's the same again. We can switch it on, you know. And the tumbling reaction is pretty interesting because the tumbling reaction is being triggered by a change of the rotation orientation, you know, of the flagellar bundle. So the flagella are only, at least for this kind of organism, forming a nice bundle that is propelling them together in some direction when the flagella rotate counterclockwise. However, when they rotate clockwise, the bundle falls apart, flows off, you know, and then the bacterium tumbles around, and then it has to wait till actually the reversal of the motor happens again, and they go again counterclockwise, and then they can go in one direction. And that's with polar organisms, with polar flagellated organisms, they can just move forward, backwards, you know, but can use the same mechanism. I do not go to this motor, just a signaling complex here, because of the photoreception, you know, it's pretty similar. We have a bacteriorhodopsin-like structure, and that is coupled or is interacting with a so-called transducer, and the transducer here has a signaling domain with a lot of methylation sites, and here key W can bind to this kind of protein and mediate it to key A. And the methylation is something for the regulation, and everything is quite well known about this kind of addiction. Just to mention you why the bacterium can see colors, different colors. It has something to do with the photochemistry. So that one here, the sensorodopsin 1, is forming a very stable sensorodopsin 1 intermediate, this 373 intermediate. And that is sensitive to blue light or near ultraviolet radiation. It goes quickly then back to the initial state. So when there's any kind of dim light, you know, the ground state is depopulated, or the initial state is depopulated, and this sensorodopsin 373 is populated. And when it sees now some blue light, because you see the reversion is very slow, when it sees now some blue light, it goes back there. Yes, you know, it's in this kind of state it was before. It's the other way around with the sensorodopsin that sees orange light. Actually, it sees orange light, you know. This one actually is being triggered by orange light. It forms a sensory complex. Yes, the sensory complex is not accumulating. It's giving the signal off. Yes, you know, please here, yes, you know, go on, you know, and then it returns in the normal way. So it's a ratio here, in this case, that is controlling the light result. That's the specialty of sensorodopsin 1. You see here, you know, it's the interaction between the helixes that is being controlled. The helix G, the helix number 7, to which the retinal is coupled, that is undergoing a conformation change, also a bit with helix A. And that conformation change here disrupts here this hydrogen bonding network with the transducer, and by that way, actually the transducer actually can transmit the conformation change to the cytosolic part that is here coupled, that is missing in this structure because it's a very long helix, you know, or helical bundle that sticks into the cytosolic part. So there's some rotation movement happening, and that is then occluding some of the interaction sites. So that's about sensorodopsin. But now, why is it so interesting for me to tell you so long time about it? Because I think it's important. What's important today in 2020? We live in year 2020, not in year 2000, is there has been discovered a fourth class of rhodopsins, so-called channel rhodopsins. They have been discovered by Peter Hegemann, a very nice colleague I know for a very long time from the Humboldt University in Berlin. I help every year. He would be a fantastic Nobel Prize candidate because he's a discoverer of generodopsins, and I will explain you why they are so important, what's actually so fancy about them. Maybe some of you have seen this kind of pictures of mice with a laser diode in the brain. Actually, they have the laser diode because he found this kind of generodopsin. So generodopsins do occur in some green algae, and that was the reason why it took him so long time to get them out. He worked for more than 10-15 years to find them, in this kind of green algae, in Chlamydomonas reinhardii. He found the generodopsins, and they are forming a channel, not a pump. So when light is hitting generodopsin, it opens up an ion channel. And a lot of ions can now go through the open channel and go into the cytosol. And when you remember how nerve pulse are transmitted, by nerve cells, it's because of ion channels. They open up because some triggering ligand opens them up. So what one is doing nowadays is one is making transgenic animals, with generodopsins, but also with phalorhodopsins. I'll tell you in a second. One is making transgenic animals, when they express a kind of rhodopsins at the right place in the brain, you can illuminate in these brain areas or these organ areas or whatsoever, and you can then depolarize the neurons and then trigger a nerve pulse going on and study, for example, behavioral change. That is cool. Halorhodopsin can be actually used for hyperpolarization, yes. That is used for depolarization, that is used for hyperpolarization because it pumps in chloride, yes. That is the way why also halorhodopsin is often used as well, the chloride pump. So you see the photocycle looks like before, tuck, 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 BR, LM, then some end state. In this area here, there is the ion channel open here between M and N, and then everything is being set back, and there's some flow happening, between his A, B, and the rest of the protein of these kind of ions. So generodopsins are cool, very cool. And we know now structures of those ones, and I think one of the problems that is still not being properly addressed is the question how one can make them more efficient. So people try by solving structures, making mutants of these proteins to get more than a few hundred protons or sodium ions being passed, because that's so far the big disadvantage of the generodopsins. They are not as superb as our natural ion channels. They let pass millions of ions before they close. These generodopsins close after a few hundred. And also another problem is, of course, the kind of light absorption and the quantum yield. So there are many things that people now try to optimize with generodopsins to understand them, how one can make them to better light triggered ion channels, because it is very important for our kind of friends who want, for example, to study in developmental biology or in neurobiology. I think now nowadays almost every second neurobiologist on this planet is using generodopsins in his or her work, yes, because this is has become a central tool for addressing a lot of questions there. So I will not go too much into the details. Most of the central residues are conserved. Only a few residues have been exchanged, like this threonine that is a bacterodopsin occurring has been exchanged by a cysteine residue for some reason, these generodopsins, and also one or the other residue. And there's, of course, also some tuning occurring. So the reason why these generodopsins are so fancy is before you had to put electrodes into brains and then activate the neurons you have hidden with your metal electrodes or glass electrodes. Now you can just use a little laser pulse, just implant maybe or use some glass light fiber to transfer light. Maybe you want also to control the color, because when you can use also color coding, you can also affect different areas, and by site-specific gene expression, you can address different areas of the brain. But here, example, simple organism, Photoreceptors elegans. Please look on this. I start the movie. You will see, I tell you what you will see. You will see there are different kind of light regimes done. This is a specialized microscope. Actually, it's possible to illuminate the surrounding of the worm. Photoreceptors elegans is a little worm, a nematode. the surroundings. It's possible to illuminate it with white light, with darkness, you know. And there's only one frame in it where it's shortly illuminated, only the worm intensely with green light, but not in the other ones. And look what's happening. So the worm is swimming around. Tuck, that's the light where the worm is in. Now it's off. Swimming around, unaffected. It was just at this single position here, you know, that something happened, because Alex Gottschalk and his people, they implanted, and I just stop here, they implanted a channel rhodopsin in the muscle cells of this worm, you know, and when it was hit by the green light, you know, depolarization did happen, and that was causing that actually no movement was anymore possible. They were arrested. So, you know, just controlling the movement of this worm. But nowadays we can have mice walking left and right, but just switching on some light in the brain, you know. So now we have even some more complicated experiments. Here's again a worm, different experiment, different transgenic line. So they raster here with the microscope, where you see this here through the whole worm till they come to a special point. Nothing is happening. I go a bit forward because it takes a bit time. Yes, you know. Go a bit forward. Now they come to the crucial area. Now it gets it. It now makes not eggs there, because they affected actually the part, you know, of the machinery where the worm actually makes its eggs and it extrudes them, yes, to the environment. So, you know, that's the way to make babies, yes, just by illuminating the right position, you know, of a little worm. So that's about some applications of this one. So we see here another kind of photoreceptor, phytochromes, with some strange music in the background. Phytochromes do occur in plants. They are important for all kinds of plant reactions, shade avoidance, flowering, germination, whatsoever. They also do occur in bacteria, in cyanobacteria, and also in non-photosynthetic bacteria. They also do occur in fungi, the phytochromes. The name is phyto means plant, and chromes means colored, you know. They are photochromic sensors for red and far red light. What does photochromic means? Photochromic means that you have an interconversion between two states by two different colors. Here with red light from that PR to this PFR state, and that one with dark red light, far red light from the PFR state to the red light. So you can switch forward, backward, just with light, forward, backward, you know. And the ratio between these light qualities tells you what you have more, this state or that state, which is actually the way how actually it works. It's a ratiometric sensor for light, and also for some of its biological reactions. So you see this here from a textbook, you know, one can see this really in the Eppendorf tube, the change of the color. It's cool, it's fantastic, yes, you know. That's also from the textbook. I don't have time for textbook. You can look for textbooks by yourself. Just the ratio determines, you know, the plant response or the bacterial response when you have it in bacteria, or the fungal response when you have it in fungi. Humans are animals are apparently the only organisms that have missed to have a phytochrome in their genome. All other organisms, in all kind of groups of organisms, you find always some organism that has phytochromes in it. Very strangely, you know, no animal uses a phytochrome for signaling. I don't know why. They are fantastic, you know. We should have phytochromes inbuilt in us. They are pretty cool. So the kind of chromophore is complex. You see, it's because the complex chemistry, I've seen it before. Actually, it's derived from biliverdin 9 alpha. That is actually what we have when we have our heme group without iron and have it cleaved, you know, by an oxygenase. Here with a diatomic oxygen molecule being cleaved here at this position, you know, that forms then biliverdin 9 alpha. Clearly there's a 9 gamma, 9 beta existing. Humans, also we also form biliverdin. These are our colored dots, you know, in urine, or when we here push our cells and get them first this red color, and then it becomes green and bluish and whatsoever. It's mostly this kind of colors from these kind of tetrapyroids. And these kind of tetrapyroids are used as chromophores here by phytochromes. So just to make this simple, phytochrome, they have two areas. Plant one, at least, the signaling one that is signaling the light, that is responding to the light, and then somehow the C-terminal ends, which are doing some reaction whatsoever, yes, inside the plant, controlling something. And the primary reaction is this one here. These proteins are very large, 100 to 150 kilodalton molecular mass in plants. In bacteria, smaller, 60, 70 kilodalton, 80 kilodalton. But you have to admit, that a change here is being transmitted to global change of a large protein complex. And I will not give you an introduction into phytochrome signaling. For this, maybe you visit some lecture, maybe by Alfred Butcher or somebody else. Yes, so when I would have time to do so. It's very, very complicated, the kind of machinery that is attached to it, the signaling machinery. But the phytochromes are the primary receptors for this, and also they're using transport to the nucleus, and export from the nucleus, and so on. I will not talk about this. I will just give you some examples what one can learn with structural biology, easily. Give you just some example from cyanobacteria, CPH1, CPH2, that we also did some time ago. They are much simpler than the plant phytochromes. You see, this is a plant phytochrome. Every dot here is some domain. Here the chromophore. Here's even something non-structured area, that is changing its position from illumination, as we know. And here's this output signaling whatsoever region. Here in the bacterial ones, it's simpler. You have just these three domains here, and you have this histidine kinase mostly. And here in this case, you have it a bit even shorter here at the beginning. Then you have two output domains, which belong to a different type here. Then again, a light-sensitive domain, and one more output domain. It's also a complicated photoreceptor, actually, because of its complex structure. Here, just to remind you, nuclear transport is also playing a big role, in plant phytochrome, but I will not tell you about it. When synthetic biology started, this was the first paper of synthetic biology that was published in Nature. It was the outcome, of I think some summer school or some activity by Christopher Vogt and co-workers, with some students, and they worked it out finally. They made a very simple arrangement, a very simple construction where they fused a phytochrome CPH1 actually, which has the histidine kinase anyway fused to it with a different kind of histidine kinase from the OMC promoter called the MZ protein, MZ and OMC. So by this they could use this kind of phytochrome in Escherichia coli and just mimic the way, how a signal is being transmitted in coli. Here you see some lawn of bacteria, yes, where they illuminated the bacteria in such a way, yes, that only those ones which are in darkness produce a black output, yes, produce a black output, and the other ones who are in light are actually are not producing this kind of color. That was very fanciful, and here's some gradients and these kind of things. So CPH8 they called it for some reason. Actually, at the beginning, this was an artifact, yes, one of the funny things. It took them time to come from the artifact because light could also destroy actually the black color by itself. They used too much light at the beginning and had the wrong construct and so on. But at the end, it worked very nicely. So this is actually the origin of synthetic biology, and it's CPH1 that was the origin. That was the structure of the photoreceptor region. So we have seen PYP before. We have seen bacteriorhodopsin before. This one looks much more complex now. You see here a chromophore that is being part of a so-called GAF domain. It's a domain type, very often found in signaling proteins. Remember signaling proteins last time. But this chromophore, yeah, it looks like that it would be like in a ligand binding site of this kind of signaling domain. And indeed, there exist some simple cyanobacteria chromes who just have this GAF domain in the chromophore. But in normal phytochromes, you have more elements. You have a pus domain here, yes, that it's also touching it here. You have a knot inside the protein. There's a knot formed for some very strange reason, a real knot in the polypeptide chain. And then you have a C-terminal domain here, this phi domain that is also touching the chromophore with its, how did we call it? The tongue, we called it, yes. The tongue, the phytochrome tongue. Like a tongue licking on the chromophore. My colleague was a fan of the Rolling Stones, apparently. So that's a chromophore, as usual, just some things. The GAF domain is interesting because the GAF domain actually is structurally very related to the phi domain. Actually, the only real structural difference in terms of the topology exists in two big loops. The GAF domain has this big loop here in orange, and you see here where the green dot is. That's actually the position where the knot is formed. The N-terminus of the path domain goes through it, yes, and crosses this loop and makes a real knot, yes. So that's of the GAF domain. In the phi domain, everything is looking here very similar. There's this long tongue, it's a different loop actually, as you know, that is actually making the contact with the chromophore. And as we do know now, this kind of domain organization in the phytochrome is not unique. We find this kind of domain organization having two GAF domains behind each other in other signaling proteins. Here in adenylate cyclase, I think it's from some bacterium. It has a GAF domain, a GAF domain. The GAF domains bind cyclic nucleotides, so there's some feedback control of cyclic nucleotide. And actually, that's one of our own phosphodiesterase 2A, but also some other phosphodiesterase of us, of animals, of humans, have also two GAF domains, and also the other GAF domain is used for controlling, or one of the GAF domains is for controlling the phosphodiesterase that is used to this GAF long helix GAF motif, as you see here, GAF long helix GAF motif. So that's actually more widely spread among signaling proteins. And the reason for that is quite obvious, there are so many convergences of structural elements here at the chromophore that you can predict already by that, yes, just by looking on it, that when something happens here, a lot of things will happen at the N-terminus, because it's also sticking here to the chromophore, despite the knot, the tongue with the C-terminal phi domain. Also something will happen here, especially because there's a conserved salt bridge being built in all these kind of phytochromes that has to be there to allow then this kind of photochromic switching, and we can predict now pretty well what's going on. And that has been also in the meantime shown by other people to work indeed. So just go on for this, I just skip here some slides that we don't don't need. Yes, again, some cyanobacteria. I like the cyanobacteria. Yes, here they migrate somehow to light. Yes, it's cool, isn't it? What about the CPH2? It's simpler. It's a simpler system. It's missing the path domain. It doesn't have the knot apparently, because no path domain here, probably not the N-terminus with a knot-like structure. It has just this phi domain and the GAF domain itself. So we analyze this. First of all, one can study that, by making a lot of mutants to remove this very complicated photoreceptor from this kind of bacteria. That has been done by Annegret Wilde. She's now in Freiburg. And one is seeing, when actually it's a very interesting reaction. It's an inhibitory reaction it has. Yes, blue light illumination, it inhibits actually the migration, yes, towards blue light. So that's one of the purposes of it, but not the only one. So I will just cross this, we will not have so much time, So that's when you work with photoreceptor proteins, yes, you see them, that's very simple, yes. Other proteins, as you should know, are colorless, okay, you can see also the colorless proteins, and you shake them, that they can become foamy, yes, okay, that is telling you that there's some protein in it, at least when there's no detergent in it, but clearly one shouldn't make foam on purpose. And one can study all the kind of things here, I think that was just a result slide, of this. So that's the structure of this protein. It looks very similar like the one that we have seen before. Nevertheless, there are some interesting points. What we found is that the tongue of this phi domain has a very special motif. It was not only the salt bridge that we have seen before, there are also two aromatic residues, these two aromatic residues. And we found that these two aromatic residues are conserved in all the sequence of the phytochrome. So you see here combining structure information with conservation. So we see the structure, we ask, is it important? We look on the conservation. We find that all phytochromes that has this phi domain have here aromats, mostly tryptophans, conserved, yes, without any neglect. And that's far away from the chromophore binding site. That one is here, yes, Here is also this salt bridge. That is easier to understand what's going on. So what's the role of this kind of tongs, of this aromats in these tongs? You just compare with other structures. You know, some people made a structure of the product in the different signaling state in the PFR-like state. We made it in the PR-like state. And by this, just by comparing the structure, we notice that actually the tryptophans, and also by making a lot of mutants, that the tryptophans are just swapping their positions, you know. So in the initial state, the one tryptophan makes tight interaction with the GAF domain, and this one is in here a bit more loopy, and there's a beta stalk region here that is making this loopy region and this salt bridge interaction. And after photo conversion, you know, they swap their positions. The other tryptophan, the other big aromat, is now anchoring everything now to the GAF domain. And also you see there are some changes in the interaction of the aspartate. It's now in interaction with a different residue. And this is actually the major central switch of the phytochromes that is allowing them to exist in two stable states without reverting spontaneously back to the initial state. So while you need then a new photon, maybe in this case, far red photon to reverse now here the isomerization state and getting this being unfolded partially, this tongue unfolded, refolded, you know, getting it now reassociated with the other aromatic side chain. That's about the other residue, about the other module, but that we can skip almost. So phytochromes are a very large family, and I will tell you something about this kind of dendrogram when I come give you the final last 5 minutes on cryptochromes and photolysers. It's my super short. Oh, what's the time now actually? I missed the time. Oh, I still have a few minutes left. So just to go back here, so we see here all the kind of phytochromes, all of them in the sequence database available, also with where we know structure, some of only a few of them. We see that the plant phytochromes clustered together with the cyanobacterial phytochromes, you know, the CPH2 that we have studied, you know, was found here in this big cluster of bacterial phytochromes. Probably the ancient class was originated somewhere here, yes, you know, in this area, you know. With these two groups that we have here assigned, you know, GAF only. Then they got a second domain, a second GAF domain by gene duplication, and they specialized it. And then they got a PAS domain and came up in this area here in this canonical regions, where there are a lot of large majority is actually being found. And interestingly, that one that we have here in gray is only found in cyanobacteria, but not in other organisms, not in fungi, not in plants. So for the last few minutes, we switch into the final class of photoreceptors for the day. Let's talk about photolysed cryptochromes. Photolysed cryptochromes are a common protein family. You see here now a phylogenetic dendrogram. Here is different proteins shown, or gene proteins shown, according to the kind of phylogenetic relationship and the distance in the phylogenetic tree. Think about as you would have your own phylogenetic tree, your parents, your grandparents, grandparents, and so on. Yes, you know. So this is actually how related they are now here in this phylogenetic tree. What you see here, it's a common cluster for photolysing cryptochromes. Here's different so-called CPD photolysers, class 1, class 2, 6,4 photolyases. Then here are some things that are called here CryPro, or we call them nowadays bacterial 6,4 photolyases. These are cry-dash, you know, plant-type cryptochromes. And you see, you know, cryptochromes and photolyases. Actually, a short definition of what actually a cryptochrome is. A cryptochrome is a signaling protein that reacts on blue light and transmitting the signal. And the photolyase is structurally highly related to this protein, but it repairs DNA. That is cool. It can repair DNA. It has a DNA lesion. I will not talk too much about that way of mechanism, maybe a short overview. So what is common and what's different? So when you look as a structural biologist to photolyze the cryptochromes, you can say when you look on different structures, and here's just a little collection of different structures from the database. Many of them are from our work, but also some other people's work, you know. It looks pretty boring because you can say these structures, when you look here for this kind of C-alpha traces, they're almost identical, yes. Also, what's almost identical is they have all the same cofactor, FAD here bound. Nevertheless, some of them do repair DNA and some other transmit a signal. And there's no way, yes, how this can be trivially recognized just by making a structured superposition. You just see that there's something in common. Also, to get things more complicated, we know nowadays that also cryptochromes existing in animals, at least, which are capable, which makes the ability to the animal to sense magnetic fields. So you see here Rotkehlchen, European robin, some bit rare nowadays. They migrate from the Africa, here North and Central Africa, to Europe and Northern Europe. And they orient themselves not only according to landmarks, but what has been shown is they orient themselves also according to magnetic fields, to the Earth's magnetic field. And now think about chemical reactions that can sense a magnetic field. That is damned hell, yes, Thinking about a chemical reaction that can sense magnetic fields. Actually, the cryptochromes have a chemical reaction that is potentially capable of sensing magnetic fields. But I think we will not have time to go into this into too much detail. That would go us a little bit away. So the central common point between all this kind of photolyzed cryptochromes is the flavin chromophore and what they do with the chromophore when light is shining on it. So these proteins use the flavin chromophore that is absorbing the light to transfer electrons. So when the FAD is in the oxidized state and it's excited, it becomes a very strong oxidation reagent. FAD-OX in the excited state is extremely powerful in ripping off an electron from a nearby tryptophan residue and forming then this kind of semi-humanoid state here, FAD dot minus. Some cryptochromes stay in this state, like the insect cryptochromes. Most other cryptochromes and all other photolyases then get somehow a proton. The mechanism on the pathway is not known yet, only some ideas. Get a proton, to reprotonate the FAD to FADH dot. So it's a radical. The radical can be very stable. It can be stable. You have protons that are stable for weeks and months in the semi-unit state when you store them in the fridge. So this state is again capable of making maybe a signal when it's a cryptochrome, or absorbing a further photon, being excited again and ripping again off an electron from the nearby tryptophan that has been in the meantime being refilled, of course. So from the fully reduced state, FADH minus, that could be also a signaling state. Who knows? Some cryptochromes, like the plant cryptochromes, can form all three states. Now the photolyases, they depend on the presence of FADH minus, because now when FADH minus is excited, FADH minus in the S1 state, it's now not a very strong, powerful oxidation reagent, but now extremely strong reductant. So with about minus 1.3 volt, it transfers its electron on anything that it's in its near neighborhood. In this case, it's a DNA lesion, and the DNA lesion gets the electron whether it likes it or not, and then decomposes by itself without activation barrier, and the electron is then is a back transfer to the chromophore, and that's it. So the big common sense point is that here we have light-driven electron transport, similar like in photosynthesis, where we also have light-driven transport, but here it's done either for signaling, conformation change, or for transferring the electron transiently on a DNA damage to repair it. So when analyzing sequences, and huge number of sequences, you have seen this with phytochromes. I want just to tell you what sequence similarity networks are. So you have heard, hopefully in the biochemistry forum, hopefully something about my database talk, that we can use a sequence, or maybe also done by yourself now. We can use a sequence and make a sequence search against the database. And we get a lot of other related sequences from the database. So when we take a sequence, we can make a search, we get a lot of more sequences, which have maybe a certain kind of e-value, in terms of the likelihood, whether it's a very good alignment or more by chance alignment, lower stringency, higher stringency. So that's what we did with BLAST and PSI-BLAST. So what can one do else? You can take all the kind of sequences where you know they belong to a certain kind of protein family, and you do for every single sequence a BLAST against all other sequences in the database. Okay, that's a BLAST versus all BLAST, yes, Takes a lot of computing time, of course, because when you have 10,000 members in a protein family, you have to do 10,000 BLAST searches, which take, of course, time. Not as it takes already one, it takes sometimes considerable amount of time. So you can do this. When you have done that, then you can assign to every single protein or every single sequence here a relationship to the other sequences just by giving here to the age, giving it ages. And then, of course, when everybody's connected with everybody else, it's boring. Then you say, okay, show me only ages, which are showing sequence similarity according to the e-value for my BLAST searches. So here 10 to minus 14, 10 to minus 28, 10 to minus 56. And this kind of sequence similarity network clustering has a very interesting function, because we know nowadays when you do so, you are clustering very often sequences together which are not necessarily related by the kind of phylogeny. The phylogeny is just telling you what was my predecessor, but not the question, am I black colored or blonde colored, with my hair, yes, which would be actually a feature, on which I could start to cluster. And one of the outcomes is of this kind of sequence similarity network clustering that clusters very often or mostly reflect subfamilies of large protein families which have the same function. And that is cool because then when you have a large protein family with, let's say, 10,000 entries, you don't know, bacteria, fungi, whatsoever, yes, you could ask yourself, do they have all the same function or maybe other different functions? And a good example is actually our example here with the photolyzed cryptochromes. When you do this with the sequence similarity network clustering, it looks like that for this kind of proteins. You see, it's not a dendrogram anymore, as we have seen. You see that we have a lot of class 1 CPD photolyses, which are co-clustering with the plant cryptochromes. So you can say plant cryptochromes in class 1 photolyses. They are very, very similar. Only minor changes apparently happen for the plant cryptochromes that they lost the ability to repair DNA. And one knows actually the difference that have happened. It's actually mostly one single residue near to the FAD. But there are then the other ones around, for example, here class 2 photolyses, which occur in animals and in plants, yes, and do repair DNA. They are very distant. The 6,4 photolyses, they are somehow quite well related, but also a bit distant. You see here the bacterial 6,4 photolyses very distant. You see the dash cryptochromes very distant. Then you even discover suddenly new subfamilies, as we have found here, new PHL, new cry. 2 new subfamilies, which have been assigned before to be somewhere here, some photolyses like genes. Nobody has seen that before. So you can predict then, you can start to use our tools, homology modeling, and analyze and comparison with the template structure, whether what this is, and then you find this one cannot repair DNA, this one could repair DNA because it has all the kind of active site residues. And you can find out by this, by combining sequence information and structure information, you can predict structures and functions. And that is something that is increasingly cool because it allows you to answer a lot of biologist questions and also biochemist questions already before having the protein maybe in hand. So that's point 1, sequence similarity networks. Point number 2 for today, one more new technique. Going back to the beginning of the talk, remember the PYP we have seen, where actually they started to make efforts to get structures, in a time-resolved manner. So how can this be done nowadays? And we will have some talks about some X-related talk about this anyway, I think from the student talk. I just introduce you shortly to the technology here, you know. An X-ray is a linear accelerator. It's accelerating electrons close to the speed of light. There's happening strange effects, the so-called Sarsi effect, that is causing the electron pulses to shorten till they have a length of a few femtoseconds. And then with the technology, as you can also use it normal synchrotrons, you can make out of the energy X-rays, which also has then this 10 femtosecond pulse length here on Sakla, you know, in Japan. And that is a laser pulse, an X-ray laser pulse with a time duration of 10 femtoseconds, with an intensity that is super high, because in this single pulse there are 6 microjoule bundled. When this kind of laser pulse, this X-ray laser pulse, hits the crystal, it evaporates or gets being blown up, you know, just explodes, because there's so much energy in it, so high energy in it. However, before it explodes, explosion takes time, and because the X-ray pulse has a length of 10 femtoseconds, nothing can happen. Remember what can happen in 10 femtoseconds? Maybe a single vibration can happen in a bond, but not more. Yeah, so actually in 10 femtoseconds, the crystal will not have changed, and that was actually the window during which actually the diffraction did happen. The energy deposited in the crystal will of course blow it up, but that happens then after the diffraction happened, because it happens also only for 10 femtoseconds when the X-ray pulse goes here through the crystal. So you make then a second laser pulse, you know, with normal light. First, you come up with the normal laser light, zacks, you illuminate the crystal, flash, and then you come with the X-ray pulse, zock, and then you get a diffraction image. And you can now record structures of crystalline matter that has been formed about 50 femtoseconds after the laser pulse has hit it. So you can do ultra fast structural studies, which have not been possible before than about 8 to 9 years. So that's what actually can be done with cryptochromes, okay, and we are the people who do this at the moment. Sorry for this, you know, Eigenlob stinkt. Here you see some structure of photoliase, you know, and you see here what happens after 10 nanoseconds. Something disappears of electron density. Actually, the bonds of this DNA lesion are clipped, you know, that has happened within 2 nanoseconds. So actually what happens now here in this time regime are the reactions happening after the photo repair has happened, and here after 200 microseconds you see that major parts of the DNA has left the active site and has been apparently swapped out, yet without being yet have become ordered. But we have more snapshots now in the meantime. So we can now make molecular movies now with our issues, you know, when we have time-triggered reactions in crystalline matter, and we don't have any kind of a problem with time resolvement anymore. Another kind of reaction that is happening, you know, in this kind of proteins, we mentioned that before, was this kind of photoreduction of this FAD chromophore. You see here that's a kinetic scheme, you know, done by some ultra-fast spectroscopists, actually with us, you know, where we could find out the kinetics, you know, how this goes on, and actually found some reason for that, you know, why the radical pair that is being formed, because here you start from the oxidized state, the FAD becomes a radical, the tryptophan becomes a radical, the FAD stays a radical, another tryptophan now swaps with this tryptophan to be a radical till the tryptophan on the surface becomes a radical. Yes, actually that's the way how the radical pair is formed, you know, and how it's actually, let's explain how this is avoided, that it's actually coming back, because when it recombines, then of course everything was useless, yes, because only when charge separation is one directional or long-lived enough, then you can use this kind of charge separation. So one can actually also follow this kind of movements, you know, and you see here a small structure change happening on the chromophore. It starts to bend actually when it's becoming reduced after a few nanoseconds, you know, and you see some change in the salt bridge, and actually you see also some effects which have probably something to do with the maintenance and the loosening of the salt bridge here. And And that is pretty interesting because it explains us quite a bit how actually also the signal, when it's living for a few microseconds, before we miss structures of states that are short-lived for a few microseconds, maybe only because before they recombine, we can now see this kind of intermediates unaffected, by cryofreezing and these kind of techniques. And here another example, a cryptochrome, yes, on which we also working from this algae Chlamydomonas reinhardti, which I have mentioned Peter Hegemann to you before already. So there's also cryptochromin, not only this general rhodopsins, yes, Cryptochrome is repairing DNA. We showed this sometime before and uses 4 residues as an electron transfer chain. And what's cool about this protein, we studied quite a bit, is it's doing a large structural change upon the change of the redox state. So this is done here with hydrogen deuterium exchange mass spectrometry done in our department, where you can just compare the accessibility of different amino acids, whether the protein is in one state or the other state, in the reduced state or the oxidized state. And you see this here mapped on the structure. So the primary data are peptide fragments which incorporate different amounts of deuterium label over time. You map this on the structure and then you can see that something is happening up here, and actually we have nowadays much better data because we can now compare the oxidized state and the semi-unit state, where actually the change is much better to be seen than here in this publication that we published last year somehow. When you use the X-ray, you get the explanation for that, because when you use the X-ray, you see what happens upon the transient reduction. The whole C-terminal ends becomes unstructured thread. Then electron density means here negative electron density that is becoming lost in the crystals in a reversible manner. And that tells you that actually when the protein changes its redox state here of the flavin chromophore and somehow gets the electron from the surface, everything breaks up here in structural terms and becomes disordered. And that explains you, like we have it with PYP at the beginning, how this protein can act as a signaling protein while being at the same time also a DNA repair enzyme. Because the DNA repair activity is not affected in the fully reduced state, it reorders again to the state as it has it in the oxidized state, or similar as it has in the oxidized state. So, and with this outlook, about structural biology and photobiochemistry and so on, and Eigenlob stinkt, das blättere ich mal weiter. I'd just like to remind about our heroes, yes, our heroes in getting these kind of issues done, yes, Hartmut Michel and Johann Deisnova, who served the first structure of a photosensitive reaction center, where for the first time he could follow on could integrate all the kind of spectroscopic information that was existing from some spectroscopists into a structural framework and really understand how these complex machines do work. And I think, that tells you a little bit why structural biology is so cool, because it has some crossover with theory. So, for example, we collaborate also with with people who do quantum mechanical calculations, really classical theoretical chemists like Igor Shapiro at Jerusalem University. Spectroscopy is super cool, of course, yes, everybody who sees colored compounds like this, and of course with photobiology, because it's some of the fun. So, and I hope till next time I have translated my membrane protein slides into proper English back again, and I thank you very much for your attention. Bye.